네, 안녕하십니까. 지금부터 바이오의약품 바이오시밀러 제조를 위한 고객 맞춤형 미디어 및 공정에 관한 세미나를 시작하도록 하겠습니다. 오늘 세미나 발표를 맡아주실 아지노모도 제넥신 주식회사의 야론 실버버그 수석 연구원님을 큰 박수로 맞이해 주시기 바랍니다.
Thank you very much. Thank you very much for the organizers to organize this wonderful conference, and also thank you for attending here. So, my name is Yaron Silberberg, and I'm a chief scientist and the head of the Customer Solution Center of Ajinomoto Genexin. I will briefly explain what you see on the screen. A few things here. CELLiST is actually our brand name of our cell culture media. OCS Lab is a lab for customer solution and customer services, customer support in Osong. So Osong Customer Service Center is a lab that we opened about 3 years ago in Osong Bio Valley, which is in the center of Korea, and I will explain about that. So today I will talk about the way we allow customers, which are biologics and biosimilars manufacturers, to optimize their pipeline and also to select the best cell culture media and customize it according to their needs. A quick overview of Ajinomoto and Ajinomoto Genexin. So Ajinomoto is basically more than 100 years old company, which is expertising in amino acids, started with monosodium glutamate back in 100 years ago, and continue to specialize not only in amino acids, but lots of other chemicals and other biochemicals as well. During the past 10 years, there was a lot of accelerated development in cell culture media, and it eventually ended in the establishment of Ajinomoto Genexin, a joint venture between Ajinomoto and Genexin, which is a Korean biopharma. This is the headquarters in Songdo, which is also the main factory in Incheon. We developed CHO cells media, cell culture media, and also we support stem cells media, which is manufactured in Japan. CELLiST is basically a short for cell specialist, combination of cell and specialist. And we hope to think that our cell culture media is specialized for making the most productivity out of any kind of CHO cell culture. There are many centers around Asia and the world, not only in Korea. As I mentioned, the main headquarters for Ajinomoto Genexin is in Songdo, where the factory is, and the support center, OCS, which is in the Osong Bio Valley near Cheongju. But we also have the main R&D center in Japan. In India and China, we are now developing customer service center as well. And also in Taiwan and Russia, we have distribution centers for our cell culture media. Now, I mentioned about Osong, so just a few quick words. Osong is quite a big biocluster, one of the 3 economic zones of Korea, South Korea. And there are more than 200 biopharma and biomedical companies only in that region. There are also the KTX line that goes through here and nearby airport, so it's very accessible. And we are located here in a center which is called SB Plaza, Science Business Plaza. And there are many, many companies, as I mentioned, some big names like LG, Binex, and others, and a lot of companies which are spin-off from university, including Cheongju University campus, which is there. In this center, we are remote assisting customers as well as working together with customers on the pipelines for biologics and biosimilars. Now, quickly overview of what does it mean, the biologics drugs, and what is the market size. So, as you can see, the global biotherapeutic market, which include original biologics and biosimilars, is over 100 billion USD, over 100 billion US dollars per year, and it's growing rapidly. So this is a really huge market. And you can just see by the amount of biologics that are expiring every few years, which means every biologic that is expired, every patent which is expired, has dozens of companies working on biosimilars for the same biologic. So the biosimilar market is really huge. And the biosimilar market needs a lot of cell culture media, which we produce. So just quick overview to show you what is the location in the whole bioprocess of cell culture media, what is the role of the cell culture media itself. This is a kind of overview of a general bioprocess to manufacture any kind of biological drug or biosimilar, like anti-cancers, antibodies, also vaccines for corona. It's quite similar pipeline, workflow for any kind of pipeline. So the first 1 or 2 months at the time where the cells are grow and proliferate and divide inside cell culture media. Cell culture media is more than 50 different components, nutrients, including minerals, amino acids, vitamins, metals, many things to keep the cells happy and allow them to manufacture the antibody they make or the protein, any kind of biologic that they are creating. So in this important step, the cell culture media basically decides how productive the pipeline is. And productive means how much tighter, how much concentration of IgG or protein that cells are producing is produced. And in this 1 or 2 months, the cell exponentially divide and produce all the protein of interest, which is later going through a lot of complicated purification steps, in which I will not talk about, but our cell culture media is mainly at the initial stage. So just a basic workflow for how do you evaluate cell culture media. This is a kind of a workflow that is employed by most labs in the world, or most centers, most companies that want to basically evaluate a new cell culture media for their cell line. We offer a few addition services, few addition kind of bonuses, which I will mention later. But basically, the important step of cell adaptation, the initial step, is quite important because it allows the cells to adapt to a new media. Why should we adapt it to a new media? I will mention later. After adapting it to the media, we want to perform batch and then fed batch culture in order to screen between different types of growth, basal media and feed media supplements, in order to find the best combination. Later on, there are more optimization steps, which are basically more basic optimization of components and process parameters. And later on, there's optimization which could be more advanced to affect or to fine-tune the protein quality attributes, other things that I will mention later. In these 2 steps, you can employ a traditional way to do basically one factor at a time, or even design of experiment, DOE, or you can go deeper into IT tools, omics approach, even artificial intelligence tools to allow you to optimize the cell culture media to your own pipeline. So just a few points, about 10 points which are really important in order to basically grow a cell culture and get the optimal productivity from it. It's an important point that I will not talk about too much because we don't have too much time, but just important, maybe most important, is the few first steps, which are recovery passages. So the cells are frozen, and we take them out of the frozen liquid nitrogen and grow them in a physiological temperature. It takes them time to adapt and to recover from the shock and freeze, right? So it's important to keep a few recovery passages before we go to the main culture for the production stage. Also, the adaptation should be taken a few stages, and I will talk about it later. Other issues which are important to notice: the density of inoculation, keeping sugar, glucose to maintain the cell metabolism, and also growth factor if the cell line require. Other issues like glutamine, optimal temperature, and pH, osmolarity. The ionic concentration is really important also for productivity and cell proliferation. and also oxygen transfer, and of course, to keep aseptic condition to avoid contamination. Now, I'm talking about media switch, and I just wanted to mention why do we want to consider media switch. There are many reasons why a company, a biopharma, wants to change from the original media to a new media. One of the important points is that the original media might not be the best media, might not be the best media, cell culture media for the cell line. It could be an old developed many years ago, and these days there are a lot of advances in making the cell culture media better. So new type of media are offered by companies like us, which are better for the cell line sometimes. It's really case by case. Every cell line prefers its own media, so you have to test and screen. Some issues, of course, are cost and supply considerations. And I highlight supply considerations because these days, bigger manufacturers, which are dominant in the cell culture media, cannot supply, cannot answer the supply needs of biopharma companies. They have lead times of up to 6 months and even more when a company wants to buy large amount of cell culture media, or even small amount. So you have to consider those issues of cost and supply chain and lead times for supply. Media switching consideration, like I mentioned, every cell line has its own kind of preference. So you have to kind of experiment and test with different media types. There's no problem usually with changing from one media to another because most cell culture media these days are chemically defined. The chemicals are quite similar, but there is some variation between manufacturers. The performance productivity really depends on the medium composition. So depends on the cell line, you can actually increase the titer, increase the productivity, double it, even triple it when you switch to another media. There are two common techniques, direct adaptation and gradual adaptation, when you change from one media to another. And I will just quickly show this table. And again, it really depends on the cell line and how sensitive it is to change of media. So you can perform either a quick adaptation step like 50-50 or 100%, which means basically using 50% of original media and 50% with the target, the new media, in one passage, and the next passage directly transfer it to the target media. Or you can add more steps, like in the table. You can do 50-50, 75, 90%, and then 100% of target media. So usually you have to check these conditions. And what we do in my lab, in my center, we usually employ several sequential adaptation procedures in order to find the most appropriate for the cell line. The criteria is to keep enough, Criteria is to keep enough cell density and also to keep viability more than 90%. And you can see in this example, this is, for example, a cell line that were recovered for a few passages. And from day 9, there was a start gradual switch from one media, from the original media, to different types of media. So we tested our media and also other manufacturers' media. So every color is different types of media. But you can see that after gradual 4 passages of adaptation, you can see that it increases the cell density. The VCD is a variable cell density, basically, and it's more than 2 million, so it's definitely okay to continue for the main culture after that. And viability is maintained above 95%. So just an example for adaptation and the importance of adaptation. I want to mention a bit about the media process for optimization. The optimization process for media to increase the media efficiency, we can say, for the cell line. Also the process parameters are basically kind of a modular process. And every company is doing it maybe slightly different, but that's kind of general stages that are being done. And we do the same. In stage 1, we do media screening, both basal, growth, and feed combination in order to find the best suitable combination, to find the best productivity, best titer. And in stage 2, we also optimize feeding manner. Feeding manner is really important, like how often you feed the cells, every 2 days, every 1 day, and the volume of feeding. So the first two stages can be done in Flask or in Amber, which Amber is a microbioreactor system. We have several of those systems in our lab, and it's a really important system to allow controlled condition of the culture. The stage 3 looks at CPPs, or critical process parameters, like pH, temperature, agitation speed, and oxygen and aeration. So those are important to increase the productivity and to maintain the cell viability throughout the culture. Later on, stage 4 is really dealing with components, so optimizing different components like cysteine and tyrosine concentrations, which are amino acids which the cells need, other supplements, and also the amino acid composition in general can be optimized. And finally, usually it's a final stage, but it's also done in parallel, is optimizing protein quality. Glycosylation and charge variant are really important for FDA or EMA approval. You need to match the original drug. If you're creating a biosimilar, you need to get the right range of glycosylation pattern and the charge variants. So I was talking about the Amber, and the Amber system is actually really useful for optimizing all kinds of parameters. So I mentioned feeding volume and also temperature, pH. We can test a lot of things, and everything is done inside the control environment, because it's the Amber system automatically maintains temperature and pH and the DO, dissolved oxygen level. So it's much more kind of realistic compared to Flask culture, shake flask, because it simulates the real bioprocess in larger scale. It's not exactly like large bioreactor, but it's a good approximation simulation. This system is also used for scaling down. After you scale up, you need to fix something, you scale down. And I should mention, it's about 10 to 15 milliliter volume size. A few other things I want to mention is related to media composition. So those attributes are quite important when deciding the components of media. Of course, of smolarity that I mentioned, the pH that should be in the correct range. Solubility is an issue because tyrosine and cysteine will have some issue of solubility. Tyrosine has low solubility, so it's usually added as a dipeptide using the feed, like glycine tyrosine. Cysteine is an issue of stability. So for example, cysteine becomes cysteine by oxidation, then it precipitates. So it could be an issue which we have to deal with. So that consideration allows to decide the composition of tyrosine and cysteine, for example, in the growth medium, compared to how much we add by supplementing in feed. The cost is really important too. So for big manufacturing, you are using large scale tons of cell culture media. So you really need to consider the cost for the process, and also the effect on overall process. For example, some component might be affecting downstream issues like purification. Some supplements could affect the column for purification of the protein, so you might have some problems downstream, so you need to consider. Safety, handling, and shipment, the availability of the raw materials is also important when you deal with the composition. Okay, next, I want to talk a bit about what you can do for optimization. How do you optimize and enhance the media? So there are kind of generally two distinguishing between two ways of doing it, trial and error, or doing it more intelligent shortcut. So I'll give you an example from maybe something you know, the Squid Game. Probably you've seen it, I hope. So Ajinomoto game, very good drama on Netflix that I saw recently. If you remember this part, if you saw it, when they have to cross the glass bridge, this is kind of example of trial and error. You don't really know, you just try. You might live, you might die, right? So this is a way to optimize the components by adjusting each component. But there are many components, right? There are, as I mentioned, more than 50 different media components. We cannot just adjust all of them. We need to consider which are more important. We need to find shortcuts for that a bit. It's actually almost impossible to cover all the combination because you have to consider synergetic effect between components. Not only one test. You need to see that one affects the other too. So for finding shortcuts is another way, basically. Like Gihun, right? His name, I think? Gihun, the character from Squid Game. He found a way to kind of shortcut this game, right? If you saw. The idea is to check the right component we want, not alter all the components. Identify the critical bottlenecks. So critical bottlenecks mean in the metabolism of the cells, which metabolic pathways are more important and less important, which components we need to target in order to efficiently optimize the media for the pipeline. So I will start with a simple idea of DOE design. Just an example. I'm not going into details because we don't have much time, I'm afraid. But I want to mention the idea of taking a few amino acids. As you know, there are over 20 amino acids, usually in cell culture media. We identify the important ones. We take 4, and we can prepare some kind of a full factorial design using DOE approach. There are 16 different combinations because you have to remember this synergetic effect between 2 amino acids could raise an exponential effect when they are both together increased, for example. So what we do in our lab, we use this system, which is quite a unique robotic powder dispenser. It allows us to accurately weight and mix every component in cell culture media. And we can make any mixture, test it on a microbioreactor system, then analyze all the amino acids and see which ones are consumed more and which ones are consumed less. And of course, check the productivity of the whole system. Basically, check the titer, the protein or IgG titer. And finally, multi-omics analysis is something I will touch later. That's really looking deeper into metabolic pathways. So that's kind of the procedure we do. And for DOE, just design of experiment, you can use statistical software like JUMP, for example, for analyzing correlations between components and the final productivity. So I'm not getting into all the details, but basically you can check, for example, that amino acid 1 and amino acid 2 have some kind of synergetic effect. Increasing both of them allows increase the titer by 20, 30%. That's quite huge when you're talking about a large pipeline production. So this allows to find combination effect between components. Now, this is a kind of traditional way to do the optimization of any kind of process or cell culture media itself. But there is a bit deeper approach that's been investigated more in the past few years, and it's called the omics. Omics is a kind of a general name for proteomics, metabolomics, genomics. The idea is to systematically, by using a systemic approach, analyze and simulate and model all the biochemical pathways, or as many as we can, inside a cell. So you're actually using something called mechanistic approach, mechanistic system, together with the statistical data analysis. And combining the two is quite powerful. There's also use of AI models, as I mentioned here, to simulate reaction kinetics, for example. Now, just quickly about the benefits. As I mentioned, there are more than 7,000 biochemical reactions are modeled for CHOK1 line, for example. And a few communities around the world gathered together to combine all the bioreactions, more than 7,000 of them in hundreds of metabolic pathways. And that allows us to really find the flux of metabolic pathways and find the bottleneck in the system to see which components in the media we need to alter and change in order to increase the productivity. We can identify components, and we can also affect protein quality. So not only productivity, but also protein quality, which means glycan profile, charge variants, which are important for FDA approval or any kind of regulatory body approval of biosimilar and biologics. So just a quick overview of how this system works. You have to get some wet data, wet experiment data, to get all the culture profile and the data of amino acids. Then it's been processed, analyzed, both mechanistic and data-driven statistical method. Then there's a design and a validation using the data and seeing how it affects the culture. So it's really general. Sorry that we don't have time to get into it, but we employ this kind of technique to optimize our cell culture media. I'm talking about this, so basically a few slides to promote my center, to let you know what we do in my center. So OCS Lab, as I mentioned, is Osong Customer Service Lab. What we do are 4 main functions. Customer support, even remote assistance, which means customer don't have to give us their cell lines. We can support them remotely. We can give them media and we can analyze the data and use also spent media analysis in our facility. But a lot of customers choose to send us their cell line from their pipelines, and then we can go adapt the cells, test different media combination, and enhance and optimize the media. We do a lot of protein quality analysis. We require high-tech equipment for glycan profile and charge variants, and we also support scale-up. So this is kind of a general workflow of how we do. First of all, we do the production of the cell culture media. We can customize it to the customers. We can culture it, as I mentioned, do spent media analysis, all the protein quality, and then do the multi-omics analysis together with collaboration we have. Sorry, I'm rushing a bit because I'm almost out of time. These are important systems which are quite unique, quite expensive, and make efficient throughput in this analysis process. So what I think makes us unique compared to other manufacturers of cell culture media are mainly these 4 things. We are given quite a personal, customer-centric approach because we are not so big, honestly. We are not, it's hard for us to compete with a huge manufacturer of cell culture media, but we can make a very flexible process service to the customers. We also make give shorter lead times for supplying cell culture media and manufacturing them. And we do a modular service approach, which means, for example, as you see in this chart, customer can go inside this workflow at any time. They can start in the middle. We do media screening, or level 2 is optimization of fed batch process optimization for process parameters. Then we go for media optimization, inside formulation changes, and protein quality. And we optimize it trying to fit the timelines and the goals of the customers. We also employ new collaboration with high-tech company which doing AI analysis and other process optimization using digital twin. In silico is one of them. They are creating a biosimulation of any cell culture or pipeline process. And as I mentioned, some collaboration both with industry and academia allow us to really dig into optimizing the culture media into specific cell lines. Quickly, this is our product, but if you want some more information, just come to me later and I can give you more. This is some results of how our media is different from other manufacturer media. For example, from Thermo Fisher and other companies which are making cell culture media. Of course, results vary by cell line, but one point that our cell culture media provide is a longevity of the process. So as you see, the red line kind of extends, and we find that our cell culture media go not only maximum VCD, but also extends throughout the culture. We are providing a really good way to allow customers to switch from whatever media they use to our media. We do it quite fast, and it's very low cost. We give a lot of resources and full documentation assistance when you apply for regulatory approval. Oops. And as I mentioned, this is the function we support. So I will quickly finish here. And I welcome all of you, if you want to come and visit, just drop by Osong Customer Service in SB Plaza in Osong. Send me an email if you want if you have any questions. And that's the end. I will finish another picture from Squid Game because I really like that show. If you have any questions, please let me know. Thank you very much.
혹시 질문 있으신 분은 손을 들어주시면 저희가 마이크 전달 드리도록 하겠습니다. 네, 오늘 귀중한 시간 내어 발표 진행해 주신 야론 실버버그 수석 연구원님께 다시 한번 큰 박수 부탁드리도록 하겠습니다. 오늘 귀중한 시간 되셨기를 바라겠습니다. 감사합니다.